So, the three labs that we're going to go over on our midterm are your enzymes, peroxidase, properties, in addition to the diffusion and osmosis, and lastly, it's your photosynthesis. So we began this course by discussing, if you remember, the very first lecture that we had when I had all of those abrupt slides, okay? I discussed with you when you have two hot slabs, one 500 degrees Celsius, one 100 degrees Celsius, you touch them together, they're gonna even out at 300 each. Then we talked about how if you have two sides, one is 500 milliliters of water, the other one is 100 milliliters of water, they're going to even out at 300. So you must understand two parts of the point. The first half is there was always going to be substance movement from an area of a higher concentration to an area of a lower concentration, provided there is a permeable membrane. If you have 500 milliliters of water on one side, 500 milliliters of water on one side, and you have impermeable membrane of 100 milliliters of water on the other side, you're never going to have diffusion. But you need a semi-permeable membrane, semi-permeable membrane, also known as differentially permeable membrane. Is going to allow for the movement of water from an area of a high concentration to an area of a low concentration, and the movement is not going to be forever, from 500 to 100 until you have 600 on one side, 0 on the other. That's not going to happen. Yes?
Would you get the same reaction if the membrane was fully permeable? It would also equal out, right? If it was fully permeable.
If it's fully permeable, it means there's no need for a membrane because it's fully permeable.
Oh.
But here is semi-permeable because that's going to have pores that allow for the passage. Pores that allow for the passage of water. So this is now comparing water. Let's say I have a separate beaker with glucose. And many students think that 500 millimolars of glucose represents how big glucose is. No, it's not like a 500-pound person and a 100-pound person. No, 500 millimolars is the quantity, like 500 people. 500 people on one side and 100 millimolars of glucose on the other side. So if you make a differentially permeable membrane that allows glucose to pass through, then the 500 is going to move to the 100 until they even out at equilibrium, and both are going to be 300 millimolars of glucose on both sides. Are we clear? Okay, now the problem is when osmosis comes in. Students have a problem understanding the difference between diffusion and osmosis. Osmosis is a form of diffusion because diffusion is the movement of particles or substance from an area of a high concentration to an area of a low concentration. But in osmosis, we are moving like water to fulfill one of the properties of water, which is being a versatile solvent. So water has many properties, one of which is it has high specific heat capacity. High specific heat capacity, and number two, has a high specific heat capacity, and number two, it is a versatile solvent. It is a versatile solvent. It is going to dissolve. So what's going to happen is you have 1 liter of water here. In both sides now you have in both sides you have you're going to have 1 liter of water. 1 liter of water. 1 liter H2O. 1 liter H2O. And here you're going to have 500 millimoles of glucose and 100 millimoles of glucose. And if you open a differentially permeable membrane that does not allow glucose to move, so now you have 500 millimoles of glucose bathed and dissolved in 1 liter of water and 100 milliliters of glucose dissolved and bathed in 1 liter of water. So the water is even, is the same. The difference is the solute. So what's going to happen now, water is going to move from an area of a low solute concentration, the 100 millimoles of glucose, to an area of a high solute concentration to dissolve that substance. Okay? Are we clear?
I have a question.
Yes.
So the glucose is not gonna diffuse from.
Because this permeability membrane is selective, and actually glucose is so big. Sodium is so tiny. That's an element. Glucose is a molecule made up of oxygen and so 5 carbons, 6 carbons. Okay? It's made up of 6 carbons. We said in the photosynthesis reaction that glucose looks like this. H2NONCN. That's all the carbohydrates look like that. But for glucose, N is a 6. So it's C6. 6 times 2N, that's 12. And 6 times N, that's a 6. C6H12O6. C6H12O6. Okay. Now, what happens now is only the water is going to move to dilute and dissolve this 500 millimolars of glucose. So that is osmosis. The other thing that you are going to be asked on the exam is what happens to the red blood cell in a beaker. So in a beaker, if we have 3 beakers, A, B, and C, and we have red blood cell on the 3 of them, and the red blood cell has 300 milliosmoles inside. The osmolarity inside is 300 milliosmoles, and the osmolarity inside is 300 milliosmoles. But for A outside, it's 1,000 milliosmoles. So that's, the osmolarity is so high on the solvent. So here, the solvent osmolarity is much higher than the red blood cell or the solute osmolarity. So here, there is so much solute outside, the water is going to go to dilute the solute, is going to leave the red blood cell, and that's going to shrink. Is going to shrink. This solution is hypertonic, hyperosmolar, and hypertonic. It's very important. Here, when you have the solvent also being 300 milliosmoles, you will have a steady state, an equal rate, because the solvent osmolarity is equal to the red blood cell osmolarity, and the solvent osmolarity and the red blood cell osmolarity are both equal. You will have a steady state. You will have a steady state where the water going into the red blood cell and leaving the red blood cell, this rate is going to be the same. That's known as steady state. Steady state. Steady state flow. Yeah, that's isotonic. Excellent. Very good. That is isotonic. This is isotonic, like an isosceles triangle. Things are equal. And you have 300 on the inside, and for example, Um, 300 on the inside, and on the outside you have 100. 100 is so little. 100 is so little, then all the water is going to go to dilute, go from area of lower concentration gradient to a higher concentration gradient to dilute that solvent, and that is known as hypoosmolar or hypotonic solution. This is on the exam. This is really important. Then we also responsible of knowing what is Brownian movement. Brownian movement is when kinetic energy is heat. Remember, temperature is known as the average velocity of molecules bumping and colliding and bombarding with one another. So remember, Brown did what he did was he had a beaker and he filled it with water and he dropped from a dropper. He dropped a solvent. He thought that the dye is going to go straight down, but the dye was moving in a zigzag line. And he discovered that this zigzag line is based on the bombardment and the collision between this dye and the solvent molecules. They're going to be colliding with each other. So the movement is not going to be smooth, okay, due to the collision. So that's Brownian movement, based on heat and kinetic energy movement. Yes, Chris?
How do you spell Brownian movement?
Brownai. Like Brown, his name is Brown, and they named it after him. Brownian. Okay, I think it's here somewhere.
Yeah, there's that one was right there.
Oh, okay.
Yeah. Okay, then we move to, you have to have a good understanding of osmosis and diffusion. Then we talked about dialysis. We said that this dialysis bag is a model for what? This dialysis bag is a model for the cell. It's representing the cell. Why is it representing the cell? Because the cell membrane is differentially permeable, is semi-permeable, does not allow for the passage of just anybody. It allows for the passage of certain particular molecules. Okay. In addition to the dialysis bag. This dialysis bag, when we put it in solution in the beaker, the dye flew down concentration gradient. So the key here is the pores. How big these pores are. They are so tiny, but they are too tiny for glucose, and they are just big enough for sodium to flow inward. So just because you have a concentration gradient does not mean you can go in. You have to ensure that the permeability is a very important factor. Another thing is the molecular weight. How big? What are the factors that influence the passage and this transition from an area of a high concentration gradient to an area of a low concentration gradient? What is that? That's how small the molecules are and how big the difference. If you have a beaker with 700 to 100, another one with 500 to 100, the one with the biggest gradient is going to have the fastest and the highest rate of diffusion. You also must remember that in a beaker that is full of water, let's say it has 9 milliliters of water and 1 milliliter of sucrose. So the total is 10. If you added 1 extra milliliter of sucrose, the sucrose concentration went up. What's going to happen to the concentration of water?
It's not going to be permanent. It's not going to be permanent.
I'm sorry?
Concentration of water, since it's more sucrose, it's going to be lower, right?
Lower. Excellent. It's going to go down. People say it's going to stay the same. It was 9 milliliters of water, and now we had 1 milliliter of sucrose. We added plus 1 more, so that's 2 milliliters of sucrose, and water stayed the same. No, it's not, because that's a solution. So the solution is like you are stepping on a scale. If you are on the scale, somebody else joins the scale with you, it's both of you now. So it's not just your weight. So here, if you add more solvent, you decrease the solute concentration in the solution. And if you add more solute, you decrease the solvent. That's on the exam. And I put it for
Add
you.
more.
If you add more of each, you decrease the other. It's not going to stay the same. The other one is going to drop. Yes?
So it's like a ratio.
Yeah, it's like a ratio. So it's going to change because that's what we're examining and analyzing the entire solution.
The solute will decrease.
Okay, then we moved into the other lab, and that was your enzyme lab. In the enzyme lab, we were looking at the properties of an enzyme. And you're asked on the exam, what are enzymes made up of? Are they sugars, carbohydrates? Are enzymes sugars, carbohydrates? Are enzymes lipid, cholesterol?
Protein.
No, they are proteins. And proteins are made up of amino acids. And we said that amino acids have an alpha carbon with 4 terminals, 1 hydrogen terminal, 1 carbonyl, that is an acidic group, alpha carbonyl, and an alpha amino, then an R group, okay? And what's gonna happen is, if you heat it up too much, you're gonna start denaturing. This is a question also, denaturing. Denaturing is when you break down the structure of that protein. So when the amino acid sequence comes out of the ribosome, it is a primary sequence, first degree primary sequence, just a string of amino acids connected by peptide bond. String of amino acids connected together by peptide bond. But the problem is, they're gonna start folding, making an alpha helix, making a beta sheet based on hydrogen bonding. Then, after we become secondary degrees, we fold in secondary degree. That is known as more hydrogen bonding, more hydrophobic effect. We're gonna have more extensive hydrogen bonding, more extensive hydrophobic effects, and that's gonna get us to tertiary degree. And then lastly, we're going to have more hydrogen bonding, more hydrophobic effects, and more ionic bonding, and we're gonna get quaternary structure. So these enzymes, they need pH, optimal pH. You mess up the pH, they're gonna start denaturing. You mess the temperature, you increase the temperature, they're gonna start denaturing. These hydrogen bonds are gonna start breaking. These hydrophobic effects are gonna start to get eliminated. These ionic bonds are gonna start to get destroyed. And you have to remember that this enzyme is so important because the enzyme lowers the activation energy barrier. So on the y-axis you have delta G. This is known as the Gibbs free energy. Gibbs free energy. These are known as the Gibbs free energy. And you will have reactants going into products. These are known as reactants going into products. And in order to go from reactants to products, you have to go up to the saddle. The saddle, that's the highest point. And the saddle is where you have your transition state complex. This transition state complex is an intermediate between reactants and product, and is right at the saddle of the horse, the top point. And what happens is the enzyme lowers the activation energy barrier, and it gives us an alternative lower activation energy that we gotta climb up to, and that's how the reaction goes faster. Your job is to know what are the effects of temperature. They lower the enzyme function, and that's how we get less product and less glycol becoming more amber brown. If we change the pH, we denature our enzyme is not going to work. We're going to have a more clear in the spectrophotometer, more clear, because we don't have a lot of dark products, because we're not making enough dark product because our enzyme is denatured. Then what are the effects of the substrate? The more substrate you add, is like you have people waiting for the shuttle bus. In the beginning, there's 5 shuttle buses are waiting. There's like 1,000 people coming out the subway. In the beginning, the reaction is going to go fast. 100 people in the first bus, 100 people in the second bus. So it's going to be linear in the beginning. But when all the buses are saturated and full, then the reaction rate is going to level off. It's going to level off. It's going to level off. It's going to reach a ceiling effect known as saturation. All the active sites are going to be saturated. Then you're going to have this kind of diagram. But if you adding more enzymes, now it's like you're adding more shuttle buses. With that one, you're always going to have a linear. Now it's never going to have a ceiling effect. If you just add an enzyme, you're always going to have a linear. The more shuttle buses you bring, the more people are going to join and the more trips you're going to have. So remember that the more enzyme you add, linear. The more substrate, linear in the beginning. But then you're going to reach a transport maximum. All of the active sites are going to be saturated. Okay? And remember that the enzyme lowers the activation energy, not the free energy. Not the free energy. The free energy is the same. It's just the activation energy between the reactant and the transition state complex, the saddle point. But the difference in energy between the reactant and the products is always the same. It does not change whether you use an enzyme or whether you don't. Yes? Ceiling effect. The ceiling effect. Yeah, it's going to level off, ceiling effect. And that is known as the level off, ceiling effect, and saturation. Saturation. Going to reach saturation. And lastly, we talked about the photosynthesis. And in photosynthesis, we talked about how we harvest the energy. How we harvest the energy of electrons. So what happens is, we said that electrons, you know, when little kids listen to the music, they start jumping up and down. People listen to music in a concert, they start jumping up and down. The electrons, when you project light, sunlight to them, they absorb that energy and they jump up, but they don't come down. So when they jump up, there are photosystems in the thylakoid photosystem 2. Photosystem 2 comes before photosystem 1. Comes before photosystem 1. And what happens is there is a reaction center in photosystem 2, and the sunlight is going to come, excite an electron. The electron is going to jump up. When the electron jumps up, the photosystem 2 is going to use that energy, like the waterfall. When the water comes down, there is a rotor blade, and it's going to spin the rotor. There is a magnet inside there. There is wires, and that's going to cause a magnetic field, and that's how you get hydraulic energy. But here we don't deal with water. We deal with an electron that is going down concentration gradient, and we're going to use this energy to funnel in and cause an influx, bringing in a lot of protons, hydrogen protons from the outside to the inside, and this is known as proton motive force. This proton motive force is going to be used to spin a rotor known as the ATP synthase, and the ATP synthase is going to make for us ATP. It's not the only thing. Second thing we're going to use, again, sunlight to harvest electron energy. The electron is going to come down. We're going to use that energy in order to create NADPH. So the photosynthesis is divided into 2 phases, the light phase and the Kelvin phase, the dark phase. In the light phase, you have sunlight plus carbon dioxide plus water, and we're going to use that to get our ATP and ADP. And then the second phase is called the Kelvin phase, or the Kelvin cycle. And what happens here is we're going to do carbon dioxide fixation. Fixation. We're going to fixate carbon dioxide to make glucose out of carbon dioxide and actually polymerize glucose to get a polysaccharide known as starch. So go and review some more and watch that important video about photosynthesis to know the difference between the light phase and the dark phase. Okay, so I'll let you go now. Go work on that study guide. Very important. And